Sounds good.
But while we're kind of like filling, I will just say that I actually have the Tusk here on my desk as well. So if we wanted to, I'll just there it is. Dun, 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 dun. So and so it has this kind of blue stain on it, which is we put on after we'd done the sampling. So it's actually just to show some of the growth structure here, more for illustration more than for any particular function. So I'm joining from the office at home. So my wife Diane is so ready for this thing to be out of our house. I cannot tell you how much she is sick. It's been here entire COVID. We have lived with a mammoth tusk.
So we are on Facebook, and for those that are just joining us on Facebook, feel free to let us know where you're from. We are just letting people join in, and we'll go ahead and start in about 3 minutes after. Hello, Grant. And we've got Jenny from Finland joining us.
You know, one thing I can do while we're kind of filling as well, I could share the screen showing the little movie of the of where the mammoth went. So reminds me of a little pinball kind of game, like tick, 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 tick.
So if you're just joining, this is just a little sneak peek. It is a map of where the mammoth went. Is that correct?
Yeah.
Okay.
I think it's an old version, actually. I see Clément, but it gives you, yeah, because the other one is labeled a little better, but it gives you the essence of what kind of got produced. So, and it's changing color there according to whether it's winter conditions or summer conditions. So, get to see where it was taking its vacation. Not really a snowbird, but oh, will it go around the eastern end of the Brooks Range? Oh, no.
So Grant has asked a question that I'm not going to ask because you will answer that in the presentation.
Okay.
Dick says hello. Dick
from Dick, how are you?
From the Netherlands. We've got folks from Atlanta, Montreal, Ottawa. And if you're just joining us, feel free to let us know where you're from, because we like to know where everyone's joining us from. And we'll go ahead and start in about a minute or so.
And you'll notice in this video as well, down the bottom, there's kind of a little clock that kind of ratchets back to on the year of life as well. So 8 years old, according to the Tusk, or 8 years Tusk age, 7.
For those that are joining us on Zoom, we are in a webinar, so you can We can, as panelists, we can see you, but you can't see who everyone else is. But we do know that you are there and joining us. So perfect. So I think with that, I'm going to go ahead and jump in and welcome everyone to our special edition of the Beringia Centre Science Talks. Moving programming online allowed us to share Beringia-related science to Yukoners, but to everyone across the world.
So while we're just waiting for everyone to join us, we do want to respectfully acknowledge that the Yukon Beringia Interpretive Centre is geographically located within the traditional territories of the Kwanlin Dun First Nations and the Ta'angkwatchin Council.
We express gratitude to all Yukon First Nations for whom this land is an ancestral home and Indigenous peoples throughout the Americas with roots in Beringia. My name is Keshah and I'm the Communications Specialist here at the Centre.
And my name is Elizabeth Hall. I'm the Assistant Paleontologist with the Yukon Paleontology Program, and Keshah and I will be your hosts and moderate today's talk. So any questions that you have before, during, after can be put into the comment section below, and we will try to get to all of your questions within the time that's provided.
We are very excited to have an elite team of scientists joining us today to present their new research on the Arctic woolly mammoth. And of course, I don't want to share much more because I'm excited to hear it from them.
So as you can see today, we have a lot of panelists. So we're going to not introduce everyone. They'll maybe give a brief introduction during the talk or at the very end. So stick around for the question section. So with that, I think we'll just begin. So take it away, Matt.
Okay, welcome everybody. And I want to start as well by just saying that this research is very much about movement across an ancient landscape. And with that said, I just mentioned that the Alaska Stable Isotope Facility and the mass spectrometers at the University of Alaska Trothidar campus that we used to conduct those analyses for the study are located on the ancestral lands of the Liaocheng people. And I want to start by acknowledging that these lands on which we conducted these analyses are the ancestral lands of the Dene people who stewarded these lands for thousands of years and continue to steward those lands today. And I want to thank them and respect their enduring relationship to their homelands. So with that said, I'm going to start the presentation. So we have a number of the panelists, the co-authors from the paper here today. Not everybody could join us for various reasons, but this is the list of co-authors, and our title is "The Lifetime Mobility of an Arctic Woolly Mammoth," and we'll come to this painting at the end as well. So I just want to give a very brief kind of overview of isotope ecology. So the central part of this is about isotopes, and I don't want to get us too kind of bogged down. I know there are people that do this type of science, but I also know, I think my mom and dad are on here too, so and they know a bit about isotopes. But so the world is made up of elements, and that includes carbon, nitrogen, and strontium. And many of these elements have isotopes, and isotopes are the same element, carbon and carbon, but they're slightly different masses. And those masses result in different kind of ratios in different things in and around our world. And food and drink can have very different proportions of isotopes, creating these isotope signatures in the food and drink. So I see Amy drinking some coffee there perhaps, and that has a very different isotope signature to the coffee mug that I have here in Fairbanks, Alaska. And you really are what and where you eat and drink. And that includes every part and piece of you. The little bit of hair I have has a very different isotope signature to the hair that Amy has, where she is as well. And that's partly due to the fact that we're drinking different isotope valued waters that make up our coffee. And that imparts a signature in all the things that make us up. And so I'm going to hand to Pat.
Well, sure. Hi, this is Pat Druckenmiller, paleontologist at the University of Alaska Museum here in Fairbanks. And yeah, is setting the stage a little bit. I think when people think of Alaska, especially if you're not from these here parts, it's easy to think that during the last ice age, which we know is a cold period in our Earth's history, that somehow Alaska must have just been bedecked in ice everywhere. But in fact, much of Alaska was ice-free, and it was part of what's known as a larger landmass called Beringia, which is basically a connection between Asia and North America. And many areas, such as the interior of Alaska, where we live here in Fairbanks, and even the North Slope, which is north of the Brooks Range, were open steppe environments, meaning they were dry grasslands environments. And they were ideal habitats for things like the star of our show, woolly mammoth, but also other organisms like steppe bison and horse and caribou. And many of these organisms perished at the end of the last ice age. In the case of mammoths, probably about 13,000 years ago or so in the mainland area, these mammoth went extinct along with some of the other organisms, the grazers that lived on these grassland environments. And it's been in fact, it's been one of the big questions in quaternary sciences. What happened to many of these animals? What was the cause ultimately of the extinction event that marks the end of the last ice age? There's a lot of big ideas. One of those, of course, has to do with about the same time mammoths were going extinct. We know that humans were one of the organisms moving across this Beringian landmass from Asia. What role did they play in the extinction of mammoths and other animals at that time? And also, of course, climate was changing and it was becoming gradually warmer and warmer. So it's easy to say, well, if you're an animal, of course you can move. And if you can move, why don't you just move away from the area that is no longer habitable? But in fact, we know very little about how many of these animals, what their range of movement was, how fast they moved, where they lived, were their ranges constricted, were they broad? And one of the ways of answering a question like that, in the case of mammoths, is by studying what we know about modern elephants, such as their closest relative, the Asian elephant. But really, that's a very poor analog. It would be ideal if we had an understanding of what one individual did during its entire life in terms of movement within a geographic region. And that's really what this study was all about.
And luckily, before our study, we had some really outstanding resources to tell us how mammoths moved around. One of my favorite movies when my kids were growing up, I mean, it's actually really terrific, and it has mammoths and other creatures moving around in response to advancing glaciers. And then there's Ice Age, the meltdown, and they move back north again. And this very much is a model that's also picked up by the Walking with Beasts BBC production as well, that has these stories of mammoths moving south in the winter and then north in the summer, and carting along little baby mammoths that sometimes kind of encounter troubles along the way. But really, I mean, these are stories and are based on very, very little empirical data. But luckily, mammoths carried around, attached to them, these tusks. And the tip of the tusk is very much the young part of the mammoth's life. And the base is the old mammoth, the day that the mammoth died. And so everything in between is a timeline of that mammoth's lifetime.
So these, the tusk, it grows continuously throughout the mammoth's life. And as Matt just said, and you can think of them as this stack of ice cream cones. So the very tip is actually the birth of the mammoth. And then there's a cavity at the base, and that is the continuously growing layer. And so for our mammoth, the base represents the death of the tusk. And so, yeah, and so here's a picture of a split tusk. And so these larger growth structures, the first order growth features have been dyed blue, and you can see them that they really do look like cross sections of a stack of ice cream cones. And we can use these layers to figure out how long each layer represents, and then how long our mammoth lived. And so the blue showed you the larger first order growth features. But the work that Greg Erickson did, he took very thin sections of the tusk, and you can see these growth layers that are daily growth bands. They're called von Ebner's lines. And using trigonometry, this is the only equation you will see in this talk, we can figure out what distance corresponds to the daily growth. And then from that, we can figure out how long our mammoth lived.
So, there's when we think about this timeline along the tusk, we can create in our minds a number of kind of broad hypotheses, ideas about what we might expect to see if the isotopes were to be a representation of the mammoth's lifetime movement. So one model might be no movement. In that case, the mammoth would be consuming the same isotopic composition in terms of its location, and you would see very little variability. Probably unlikely we'd see something like this, but I guess it's an extreme end member. Another model we might expect to see is very regular up and down along a timeline that might kind of indicate something along the lines of a seasonal migration. An alternative model is some kind of combination of periods of low movement or no movement, followed by like significant movement. And so we might label this a kind of more of a dispersal model as well. But these are what we had going into this. But of course, we didn't know what we were actually going to see. So
Bataille at the University of Ottawa, and I got involved in this project particularly because I'm a specialist of strontium isotopes, and strontium isotope have been one of the key tools that we've used to really reconstruct the entire mobility of this mammoth. And so you have this mammoth, and it's roaming on the landscape and eating plants, and under the plants there is a soil, and under the soil there is a type of rock. And so what we know about strontium isotopes is that they vary on the landscape according to the type of rock and the age of rock that's present on the landscape. And so you can see here, the yellow, the purple, and the red represent three different types of rocks, each with different age, each with different type and history. And each of those will have a different strontium isotope value. So as the rock are changing across the landscape, this creates pattern of strontium isotope not only in the rock, but then transmitted to the soil and ultimately transmitted to the plants. And so as the mammoth is roaming and eating plants on the landscape, and as its tusk is growing continuously, it keeps incorporating some of the strontium isotopic signatures that are specific to the location where it's roaming. And so what we did is then we've been able to relate the strontium isotope in the tusk with map of strontium isotope variation on the landscape that we predicted across the entirety of Alaska. And so this is how we did that. We did that we went and collected local rodents from all across Alaska, hundreds and hundreds of rodents from all across Alaska. And each of those rodents were collected in different sort of geology, different sort of climate condition. And so what we could do with this, we had all those points with strontium isotope that we analyzed. And then from all those points, we extrapolated a map based on the geology to predict strontium isotope now not only at those local points, but across the entire landscape. And so once we had this map predicting strontium isotope across the landscape, we could start to compare it to the strontium isotope on the tusk.
Right, and so at this point then, back to our mammoth, it's time to go tusk shopping. So in order to answer the question of understanding how an individual mammoth might have moved across the landscape, there's a couple of things you might want to look for in selecting an ideal specimen. And first of all, you want an adult. You want something that has good data, that we know where it actually was found, meaning also where did it die. So we have this one important point. Not all of the specimens we have in our collection have good geolocation data, but this is an important thing we need to know. And also you want a specimen that is in good shape, and ideally it's more than just one tusk, but part of a skeleton. And in fact, that's exactly what we looked for in selecting this particular individual. It was found from above the Arctic Circle, and the skeleton was unusual in that it wasn't just a single isolated tusk. It was actually part of a partial skeleton that included bits of the skull. And importantly, it had both tusks. So the left and the right tusks were preserved. They were in beautiful shape. They're about 2 meters long. And we were able to then radiocarbon date the material at being around 17,100 years old. And this particular individual had a tusk that suggested a minimum age of about 28 years. So it could be a few more years older than that. They sometimes wear off a little bit of the tusk at the very far end. And this particular specimen was located in an area of Alaska we call the North Slope. So it's the area north of the Brooks Range as it kind of slopes down to the Arctic Ocean. And two of our colleagues, co-authors on the paper, Dan Mann and Pam Groves, were involved in the collection of this specimen back in 2010. And you can see here collecting along the river can be a bit of a wet struggle. But this particular individual was nicknamed Kick because it was found along a tributary of the Colville River, a major river, and it's called the Kikiakarak River. So it was shortened up to Kick is the nickname for this individual.
Oh, that's me. So we were fortunate to get a little bit of the tusk back to UC Santa Cruz to our lab, where we were able to take it into our super clean room, grind it up into a fine powder, and extract ancient DNA. And this allowed us to do two different things. First, there was a lot of DNA in it for stuff that's old. Normally, you don't get very much DNA at all, or most of the DNA you get is microbial. In this case, we were lucky. We had enough that we could piece together a whole mitochondrial genome. This is a type of DNA that's inherited only down the maternal line, and it allows us to assign with really strong confidence the maternal lineage of this mammoth to all the other mammoth maternal lineages that are out there. And we found that this lineage, this mammoth, is closely related to or in the same group as the last lineage of mammoths that we know to have existed in Alaska. So that was pretty cool. Does the next slide talk about the sex, or should I just talk about that now?
You can talk.
Oh, yeah. And with the nuclear DNA, so not the mitochondrial DNA that we were able to get, we were able to say that the individual was male. And this analysis relied on a quirk, a really straightforward quirk of DNA, and that we know that we have in our chromosomes, we have a whole bunch that are autosomes, where males and females have, where you get the same, you get one from mom and one from dad. So males and females will have the same amount there. But the sex chromosomes, you're either an X or you're an XY. And so what we did was we asked, okay, how much DNA maps to these autosomes where you have two copies of everything? And then how much maps to the sex chromosomes? And if it was female, we would expect the same number to map to the sex chromosomes as the autosomes, 2 X's, 2 of every autosome. But in fact, we found half as much DNA mapping to the X, which told us that they only had 1 X, which meant it was male. So pretty straightforward, but still a kind of fun analysis to be able to do with ancient DNA.
Yeah, so in order to do a really cool study like this required a lot of different talents, and I think really have to take our hats off to the people that made it possible to actually take this tusk and split it lengthwise in order to do this detailed and very careful isotopic analysis along the entire length of the tusk. The outside of the tusk is a little hard to read. It's liable to have contaminants and impurities. So it's important to sample the inside of the tusk where you're getting a very pure and easy to read record. And so some of our colleagues at the Geophysical Institute here who are in the machine shop were able to set up a very complicated rig in order to push this tusk through a saw in order to make a very clean cut along the length. And if you've ever picked up a mammoth tusk, it's not straight. In fact, it's got a very interesting and complicated twisty curve to it. So this is no small matter that they were able to pull this off in order for the sampling then to be done along a nice clean surface right down the midline.
So once the tusk is split, it's 1.7 meters long, so it's a little too big for our instruments. And so basically, we can see that pulp cavity that we talked about earlier. And we really wanted to make sure that we were getting the very center of the tusk. And so you can see these sort of, there's the curved line all along the center, and then there's these hatch marks. So those are the 5 centimeter chunks that we took out to be able to fit into our laser ablation setup. And so we basically did like an angled wedge, and then we tried to sample as close to the center as we could to have sort of a nice continuous record. And in order to do that, we used a laser ablation multi-collector inductively coupled plasma mass spectrometer. And setting up that analysis, we had a lot of help from Johanna and Thomas at the University of Leoben.
Yeah, so we had the chance to come to Alaska to share our knowledge and experience with that type of methodology because of our long experience. So we're analytical chemists from Austria, came to Fairbanks, and this is what we found. So you see a nice picture of the lab in Fairbanks with the mass spectrometer that is being used for the strontium isotope ratios, and this type of machine allows to do very precise measurements. So one needs to imagine that the differences in the numbers that you're interested in to really track the movement of such an animals are very small. So you need sophisticated instruments to do that.
So as a matter of fact, on the left-hand side, what you see, it's not the coffee machine with which Matt produces the coffee to keep himself and Karen awakened, but this is the laser. So basically, there's a laser system which produces a fine laser beam of the diameter of 40 microns in order to ablate material, which is then directly transported into the mass spectrometer. And considering we have a task of 1.7 meters, Karen has shown already given you solve the riddle how to get a 1.7 meter task into the machine, but she will also explain you how to get the resolution of 40 microns, which is half the width of a hair on a 1.7 meter task. Karen, please.
So basically, the program that we use is sort of to do the analysis is sort of like you can think of it as a glorified version of Microsoft Paint, and so we draw the lines and focus where we want it, and then we set this laser beam to go at 15, and it's moving along the transect at 15 microns per second, which is a very, very long, very, very slow. And it takes a very, very long time to do this. And we ended up getting over 400,000 data points.
Okay, so after all of that incredible work that went into collecting this isotope data, how do we actually use it to develop a model for the mammoth's mobility throughout its life? So the way that we proceeded was we tried to identify journeys that the mammoth took that are consistent with that isotope data. So from a modeling perspective, the mammoth's life journey actually begins at the end of its life at this location of death where that tusk was found. And from there, the model makes a sensible guess about where the mammoth likely was a week before his death, and then a week before that, and so on and so on. And so what constitutes a sensible guess? So we have 2 pieces of information. We've got these painstakingly collected isotope measurements along the tusk, and we also have distance information. So we want to incorporate the fact that our mammoth won't be traveling from one end of Alaska to another in just a week. We also wanted to preclude significant changes in altitude, basically mammoth cliff jumping, as well as glacial traverses, because if you've ever been on a glacier, you'll know how hard it is to walk on, even without a 1.7 meter tusk. So for every possible location that the mammoth could go, we gave them each a score. So that score incorporates both matches to the strontium isotope data and assigns higher scores for shorter distances traveled. So that model is more likely to choose locations for the next step in the walk that has a higher score. And so from there, once we have a walk that's consistent with this strontium data, we can rank those walks using the oxygen isotope data. So Matt, if you wouldn't mind going forward a couple of slides and a couple more, please. And again. Thank you. So what we've shown here is the top ranked walk. And so that's the top ranked walk with respect to both the strontium data, the oxygen data, and the distance traveled data. So in panel A, you can see the areas of movement that the mammoth likely took as a juvenile, so traveling with its mother. In panel B, as an adult between about 16 and 26 years. And then panel C shows areas that it was in the last 2 years of its life before it, we show some signatures consistent with nutritional stress. So darker lines reflect more time spent in each of these areas. Next slide, thanks, Matt. So finally, I just want to really emphasize how critical all of the work that, that Karen and Matt and Patrick did in order to collect the isotope data. So what I've shown here is potentially two random walks. So on the left, we have the measured strontium isotope ratio along the Tusk in blue, and then our top walk according to our model in black. And then on the right, we have a panel where we have, again, the strontium isotope ratio along the Tusk in blue, and then a random walk in black. And so that walk on the previous slide shows a walk that is not at all informed by isotope data. And you can see that what we see is a walk that very poorly tracks the Tusk signature that we actually saw. So on the right is the best we could do if Matt and Karen and everyone didn't go to all of their trouble. And you can see on the left how much better we can do after all of that work. Over.
So this is just the paper, the figure that was taken out of the paper there, and just kind of summarizes the different regions that the mammoth used during those different life stages there as well. So you can see the white mammoth is the point that it died, and the gray area is where it moved in the last year of its life, and then the black squiggly lines everywhere are the route that it took between those kind of what we call frequently used areas, which are the colored areas. Also, we put onto this map the white dots and the green dots. The white dots are where other mammoth collections have been made in Alaska, and the green dots are some of the older than 11,000-year-old archaeological sites as well. We just wanted to put that in some kind of, put the mammoths movement in some kind of like context. And so one of the things that we kind of is interesting to do with some of the modeling work that Amy and Clément did was that you can also take those routes that were over this kind of GIS layer, and you can say, well, where did it move? And this is actually one of the figures from the supplementary document. So there's a huge supplementary document that went with this paper. When you produce a paper like this, you boil it down to like this much. And there's a massive supplementary document with some really neat stuff in it too, if you're really keen. But one of the things you can do is say, well, were there changes in elevation during the lifetime? Because if we know where it was spending its time across this landscape, you know, were there changes in elevation? Because obviously there's much more mountainous regions, and was definitely spending a lot more time in and around and crossing the Brooks Range, the western end of the Brooks Range, which, as Pat said, is like, you can see the white areas are glacial extent as best we know it in Alaska. And in the Brooks Range, it wasn't entirely covered with ice. There was still plenty of regions for this mammoth to move around in, and can move around that that western end of the Brooks Range, or the eastern end too. So one of the other isotopic systems, so we've really focused on geolocation, trying to figure out where the mammoth kind of moved, but we also measured a number of other isotopic systems, including the carbon isotopes on the inorganic fraction, the carbon isotopes on the organic fraction, and we also measured the nitrogen isotopes on the organic fraction as well. And you can think of that as very much geared towards diet. It's showing us something about nutrition of this mammoth over time. And I guess one of the things that really surprised us, and we analyzed these like way early in the system compared to the strontium isotopes, which took forever to do, but we generated these data a lot more swiftly. And the tip is the young mammoth again, just to orient, and base is when the mammoth died. And I kind of like covered up the end because I want that to be a surprise for you as well, because what we saw was this, which was like, yeah, it really, really surprised us. It was this very kind of distinct kind of uptick in the nitrogen isotopes. And that initially kind of perplexed us. Was like, well, that's really very unusual. But we do actually see a very similar pattern in other animals that are experiencing nutritional stress, and that includes humans, actually, includes ground squirrels, and it includes bison as well. And this is some of the work of Juliette Funk, who was here at UAF as well, studying the Anoka Flats bison. And obviously they don't have tusks to analyze, but they do have tail hairs. And so this is some of the tail hair analyses of a number of individuals that actually experience a period of very distinct nutritional stress during the end of one particularly harsh winter. And you see in this case, the nitrogen isotopes in the tail hairs kind of do a similar kind of uptick as well. And so this is something that we've seen. And using the kind of the timeline, we can say something about when that started to occur. So it was about 200 days or so, certainly within the last year of life of this particular mammoth. Started to see very significant nutritional stress, and probably this equates to eventual kind of starvation signature. And so really the end of the life is kind of a sad story, because the gray area here is the region that it spent in the last couple of years worth of life. It really kind of like cinched down where it was traveling. It started spending much more time around the area. The white mammoth is where it eventually died. And it was much more limited mobility. Using the oxygen isotopes, we could say something about not only geolocation, but it could help us inform the season as well. And it was definitely kind of wintry, spring kind of conditions as well. And using the nitrogen isotopes, we used to show that it probably starved to death. So it's a combination of all these lines of evidence that individually help kind of flesh out the entire picture. And we're going to hand to either Jeff Rasic or Ben Potter, just to place some of this in a broader kind of context as well.
Yeah, I can go first. So yes, this is of course very exciting with respect to helping understand the human-mammoth interactions. Now, mammoth is 17,000 years old, so it's a few thousand years before humans are coming into the region, but that's pretty close. So it's in the deglaciation period. We've got humans in Siberia at 15,000, and then into Alaska around 14.5, maybe 15,000 here. So I want to couch this in the context of what we don't know. So the exact nature of human-mammoth interactions are really ambiguous in a tantalizing way. Some have argued that maybe we're looking at just scavenging. We find a lot of tusk material in sites without other bits of mammoth that suggest that they maybe weren't predating them, but they were more likely just collecting scavenged ivory for use in their tools. And people like Dave Meltzer and Don Grayson and others have argued that humans weren't really hunting mammoth on a regular basis. I think the argument was maybe they killed once in their life and then talked about it the rest of their lives. On the other hand, people like Nicole Wagsbach, Todd Suravel, and others have argued that early Beringians, early Clovis peoples further south were heavily predating mammoth. That was a high-ranked animal that really enabled some of the high mobility that we think we're seeing with early humans into the Americas. So our results here are pretty exciting because they suggest a couple of different things. One, we see long distance mobility and particularly seasonal mobility in very similar ways that we see with modern caribou. Now we know that caribou are the primary large mammal that were being hunted in the Holocene in Alaska. And this suggests that the more predictable mammoth movements would be, particularly along these travel corridors between seasonal patches, the more attractive they would be to human hunters. In other words, more predictability, the more you can plan, particularly the nature of these early toolkits suggest high residential mobility, a focus on some of these large mammals like bison. And mammoth, from this paper, it suggests that it might be more attractive than we thought. We really had not much data on this. If we can turn to the next slide. So there are some smoking guns that we do have in the record. This is a picture of a mammoth vertebrae from the Lugovskoye site in Siberia. And you can see the edge of a projectile point, a microblade armed armature, probably from an atlatl guard, that's embedded in the side of this. And I think given the data that we've got prior to this from the archaeological record, we can't disentangle the climate effects versus the human effects, but it does suggest they might have worked in tandem. If we have reduced mobility as the grasslands were being fragmented at the end of the last ice age, you have potentially restricted mobility, that certainly could put stress on the mammoths and also enhance the potential for human overpredation as you have humans coming into the area with very effective killing devices. So there might have been an interplay between sort of the human effects and the climate effects at the end of the last ice age.
And I just want to see if Jeff would like to say anything further.
That covers it pretty well. The only things I would add, the thing that really jumps out at me when I look at the map, the final map, is the periods of the mammoth's life where it was focused in a relatively restricted area. It had great movement across its lifetime and at certain times of the year, but at other times, for months on end, it seems to be focused in a pretty small area. The imprint mammoths had on the landscape was tremendous. They would have left trails and very visible signs for hunters to key in on. And during those times of restricted movement would have been an especially enticing target. These hunters, we know from the archaeological record across greater Beringia and shortly after this particular mammoth lived, were highly capable hunters. So they had sophisticated weaponry, and combined with this picture that's emerging about the mammoth's behavior, that this really sort of sets the stage for that that scenario where people were, even if they weren't specialized hunters of mammoths, this was probably a regular menu item for them, rather than the scenario Ben mentioned where hunters may have encountered a mammoth once in their life and then spent the rest of their lifetime talking about it, or only making use of mammoths that were scavenged. Those seem like less viable scenarios given what we've learned here from this study.
Thanks, Jeff. And so I just want to kind of wrap up here, just finish off, and just by mentioning this beautiful painting that ended up appearing on the front cover of Science, but actually took about 2 years worth of time to produce. It was a truly mammoth piece of artwork in its own right, because it's actually a painting that was created by James Havens here in Anchorage, Alaska, and took him about 2 years to paint, and it's actually full life-sized. And the painting is actually going to be going up here in the University of Alaska Museum of the North. They're just trying to figure out how to fit it in, because it's a monster painting and really is very, very beautiful as well. And one of the things you don't see on the cover page, because they cropped it, is down in the left-hand corner, you can see some little caribou kind of following the pathway being left by the mammoth at the western end of the Brooks Range. And this is a region that mammoths aren't using today because they're extinct, but the Western Arctic caribou, some of them are still using today as well. And so we wanted to kind of put that in that painting. And James worked with a number of us that were on the paper to help inform this this painting. It's a really beautiful piece. So Clément, do you want to just do the final slide here?
Yes, we wanted to also dedicate this paper to the memory of Sean Brennan, who passed away in early 2020. He was a good friend of mine, and he was a grad student of Matt Wooler, and he really kind of pioneered a lot of this work on strontium isotope, and we were working hand in hand on on developing some of these strontium isoscape with him, and particularly published a work on tracing salmon migration using orthoids. That kind of was a background for our work. So really wanted to put a word for him, and we definitely miss him very much.
So with that said, I just want to thank you for listening. I also want to thank the panelists for joining us today as well. I think the fact that we were able to tag team a presentation, which is not typically the way that most scientists, is usually a single show types of thing. But I think this kind of seamless way that we kind of went between each of us is a mark of what a great team effort this really was. We really worked very closely together, and I think that's reflected in the presentation today. So I just want to thank you all, and thank the panelists as well.
Good job. That was amazing. So I think we're going to open it up for questions. I do have a quick question first. I'm just wondering about the morphology at the Tusk. So it's about 1.7 meters long, and you're sampling the dentin for the strontium. Was there any cementum on the outside? Did you still have any enamel on the tip?
Pat, do you want to take that?
I can try to answer that. I mean, to my knowledge, I'm not aware that there was any enamel remaining. It was all just the dentin that we sampled.
So I'll go ahead and jump with a question from Facebook. And Jenny is asking, how much of the woolly mammoth's movements were restricted by geographical boundaries?
Sounds like one for Amy or for Clément.
I mean, certainly because of our framework for modeling, it was definitely blocked by any mountain range and ice cover. So like when you had a big ice cap on the Brook Range, it was definitely not used for travel. So mammoths had to go around this pass. And same for like big mountain range were not like favorites in terms of movements. So you can see on like a lot of the movement, it's moving really around the Brook Range and kind of avoiding this area as much as possible. So geographic barrier definitely have a big impact there on the movement, but I think that's kind of realistic in terms of what the mammoths actually did. So.
Yeah, I think that's a great answer, Clem. And I would also add to it that one thing that I didn't mention that we did bake into our model is that if the mammoth begins on a walk and ends up somewhere where there's no plausible next step, so maybe it has to like jump a cliff or like leap across a glacier, or the strontium isotope for it going backwards just doesn't match what's happening on the Tusk, we actually said that's not a viable walk. And so actually of about the 20,000, we actually did about 20,000 walks, and only about 5,000 led to a plausible match for all the data. And so I would say that what that tells us is that actually these geographic constraints are giving us quite strong information about where the mammoth likely went if about 70% of our walks are being ended because there's no plausible next step. So great question, thank you.
And even out of the 5,000 walks that are plausible, only really a few hundreds actually were fitting with the isotope constraint, with what we calculated. So
yeah, a lot of sophisticated modeling constraints going on here.
Yeah.
Cool. The next question is from Grant Zazula, who unfortunately couldn't join us today. He would like to know, and I think he's actually inviting you to Matt and team, are you planning on expanding this to more tests, more localities? Would you like to come and sample ours?
Yes, and yes. I mean, Pat and I haven't talked about this, but like, Pat, I think we should split some more Tusks. I'm not going to put him on the spot. Oh, he's nodding. That's good. So, yeah.
And actually,
so definitely sampling from, and so we've worked very closely with Grant Zazula and Elizabeth and the Beringia Centre to kind of do other work on bison and horse and the like. And so I think it would be fantastic to expand this out to more Tusks.
And I would just add too that one thing that was really important about this particular specimen is that it was found on public land on part of what's called the National Petroleum Reserve Alaska, which is managed by the Bureau of Land Management. And so we at the museum here, we hold specimens that are federally owned in the public trust, and we steward these for the public. And what's nice about that is because of that, then these specimens are available for cool studies like this, where we can tell some really amazing stories about Alaska's history and share those stories. And we definitely have plenty more tusks and other cool specimens from federally owned lands all across Alaska, actually. And when we go about doing a study like this, we make sure we get permission before we do these samples. But we've had really great cooperation with our federal partners who make this possible.
So the next question is from Stephanie, and they're asking, did you look at the cortisol in the last years of Kik's life leading up to the sea spike? And then I'll follow up with that question of, was there any other analysis done on the Tusk other than the ancient DNA and the strontium analysis?
Yeah, so we analyzed a number of different isotopic systems along the Tusk: carbon on the inorganic fraction, carbon on the organic fraction, the nitrogen isotopes on the organic fraction, the oxygen isotopes, and the strontium isotopes as well. And then we also radiocarbon dated the Tusk. We also radiocarbon dated the mandible that was found with it as well. They were within error of each other, as you'd expect if they were from the same individual. I'm aware of folks that have measured cortisol in hairs before. We didn't do that on the Tusk. I'd be surprised if there was cortisol still preserved in the Tusk itself. So, yeah, I think that pretty much sums up the analyses that we did on the Tusk.
I'm just curious if you did the strontium on any of the other elements, like the mandible or part of the skull. No? Okay.
Yeah. We had our work cut out with the Tusk.
We have a fun question from Sue Hewitson. Did anyone calculate how far this mammoth would have walked over its lifetime estimate? Seems far.
That's for Amy, because she did some fun things with those numbers.
Yeah, about if we straightened out all those line segments that we see on the mammoth bouncing around Alaska, they add up to about 72,000 kilometers, if I recall correctly. So which is pretty amazing. So we'll also keep in mind that these are sort of movements from one point to the next. So they're likely maybe an underestimate actually of how far the mammoth traveled. So it's possible that even though that number is staggeringly large, the mammoth traveled even further. This is also in its relatively short, approximately 28-year life. But in the course of a typical human 80-year life for a high activity person, they might also travel around 72,000 kilometers. So there is some parallel to sort of lifetime movements for this mammoth versus lifetime movements for your modern active human.
And that equates to about almost twice around the Earth, which is kind of remarkable to think about.
So I have a question about must, and is a documented occurrence in modern elephants. Would it be possible to see in woolly mammoth remains? I think I pronounced it right.
One of the interesting features in all of the isotope data that we saw was that about halfway through the record, at about kind of 15 or 16 years within the Tusk record, we actually saw a lot of the isotopes kind of very significantly changed. And you actually saw it in the presentation with it spending a lot more time at higher elevation after its midpoint in its life. And that initially was a little perplexing, and I think it was prior to us getting the results from Beth and the ancient DNA figuring that it was a male. But once we had the designation as male, things started to make sense a little more, because as we read more about modern elephants, we know that in some modern elephant populations, males are encouraged to leave the matriarchal dominated herd to lead a more solitary kind of existence, probably for obvious reasons, but or as little groups of males. And it's after that point that they start to put on a lot more ground in terms of their movement and distance, as they seek out the resources to support them, but also to seek out reproductive females as well. So that, I mean, it probably doesn't fully kind of answer the question, but it was an interesting kind of feature that we saw in the Tusk.
Cool. So Greg McDonald would like to know, could this strontium analysis be applied to the preserved horn sheaths at Thyssen priscus?
Yeah, it almost certainly could. I know that there are folks that are kind of are working on that a little bit, and some of the tricks that they have to get around are the potential for contamination is greater, because it's a so the chance to you have to separate that out a little more, because you're dealing with keratin, which is a pretty kind of closed system. Chemical system is pretty kind of robust and tight, but yeah, there's some method development stuff that would need to go on.
Can you?
Adding on to that, the tusk is really nice because it's a mineral called apatite, which has calcium, and so the strontium substitutes in very readily for the calcium. Whereas when you have a horn sheath, it's a sort of a protein matrix, and so you're dealing with much lower levels of potential strontium being able to make it into that sample. So it makes it more analytically challenging.
And Thomas and Johanna would point out probably that, we're dealing with measurements that are made better by that concentration. We're measuring the isotope composition, but it's very much tied to the concentration. And if you're analyzing something at low, you guys can.
Yeah, as a matter of fact, this is exactly the point. I mean, the general answer would be yes. I mean, if it's feasible, anywhere you find the strontium and it's incorporated incrementally, then you can do it. And we also observe a steady improvement of the instrumentation. So sensitivity-wise, so it's getting better and better. And as a matter of fact, the answer still remains yes. So we do the same stuff on fish otolith. It can be used on horn. You use it even on human teeth. Okay, here again we go for apatite, but we are looking at very, very high resolutions and very low concentrations. And that's what we love as analytical chemists, is the analytical challenge.
Mm-hmm.
I think, Jeff, you answered this question in the chat, but I am going to ask it. Brent asked, could the mammoth have been staying to the north to avoid human predation?
My quick answer to that would be that, even though we don't have direct evidence of people in Alaska right at this particular time, they're present across greater Beringia at this time and earlier, and they're present at pretty high latitudes. So I don't think moving farther north would, if you're a mammoth, would get you away from people. People were widespread across Beringia at this general time period, and highly mobile.
Okay, Andy Padilla, is that right? Would like to know, were any seasonal patterns observed in the data that would suggest areas of breeding or other life history patterns?
So I'm going to answer this with Amy, I think, but just first a disclosure on this, and I don't know if my connection is good. I keep seeing like unstable connection signal. So if I'm cutting off, can you please stay on? But yeah, so this is a great question. And this is a question that the reviewer also asked about seasonal mobility. And I wanted just to say, first of all, that we wanted to keep our results as conservative as possible for this particular paper. So we try to stay away from the seasonal interpretation as much as possible. The reason being that our age model is not in terms of like figuring out each seasons. And so we didn't want to over-interpret seasonal movement. That said, we do see seasonal movement in the mammoths, and particularly north-south movement in the core area in particular. Those are not like, Andy, I think you were asking if you could find like breeding zone. We can't really identify like key breeding zones that will be useful, but we do see like regular seasonal movements in these individuals. Not sure. Am I cutting still? Can you hear me?
Mm-hmm. We can hear you.
Okay, sorry about that. Amy, do you want to complain about this?
Nope, that's perfect. Thanks, Glenn.
So just kind of follow up, Dick Moll is asking if you considered studying a female tusk from the same area, and do you think that the results may be the same or somewhat different, or?
Yeah, so I think there's this is related to that question of like what next, and so I think there's so many cool things we could do. For the paper, we dated a whole bunch of other tusks as well, some of which were infinite in age, but a number of them weren't. So an obvious question is, you know, if you went, ours was 17,000 years ago, what was the behavior for a mammoth that was living 30,000 years ago, or at different time points? But an obvious other question is like, well, what were female mammoths doing as well? Same or different? I know that in your collection, Elizabeth, over there, I think you have an adult tusk alongside a juvenile tusk that was found kind of together as well. So, you know, you could do some really cool things. I mean, presumably if they were a herd, you would expect them to kind of like be tracking each other as well, which would be kind of elegant and really kind of beautiful to see. So yeah, females, next question. I mean, there are many more things to do. So
we do actually have a female tusk that's already been cut. I showed it to you the last time you were here.
Perfect. Yes. Let's do that one.
Okay, easy. All right, I have another question from Greg McDonald, and he would like to know if you can tell how long the mammoth nursed and at what age it was weaned.
Yeah, I think you know, in that long supplementary document, we didn't draw attention to that in the main body of the paper that much, but it did seem like there were a number of isotopic signatures that were consistent with other papers that have identified weaning in other mammals using teeth and tusks and the like before. So it seemed like we had some features that still retained a kind of a pre-weaning to weaning kind of signature there in the juvenile part of the tusk. So I think that was both in the carbon isotopes and the oxygen isotopes as well. And I think the nitrogen isotopes as well kind of showed a bump at that time too.
So the next question is from Pete, and he asks, did you see any time or space jumps near the coast that might suggest the mammoth traveled onto a now submerged land bridge?
Yeah, I mean, there is a lot of walk that are constrained by the coast. So that's definitely a big uncertainty of this work. What we try to do is really reconstruct general pattern of mobility, and from all those walks that were following the isotope constraints, we just kind of draw general patterns that we saw from those and general mobility history. But yes, there is a big uncertainty in this work, which is we did not reconstruct the strontium isotope of the land bridge, so the mammoths would certainly have roam in this area. But what we have concluded from our simulations I think is conservative in terms of the range, the regular core areas, and these very long distance movement that we see post adolescence. So I think there is more work to do on this to try to maybe reconstruct the strontium isotope of the land bridge and do a little better in like maybe constraining the exact path. But at this point, this is the best we could do.
Yeah, just to add to that as well, it's a little tricky to kind of create a strontium isotope map for the Bering Land Bridge because most of it's underwater right now. It got submerged after the end of the last ice age by rising sea levels, and so I mean just to mention, I mean one of the things, so we have a new project just starting, which actually has us going out into the Bering Strait and taking cores down to get parts of the previous landscape that was the Bering Land Bridge when it wasn't submerged underwater as well. And so one of the things that we could do is measure the strontium isotope compositions to at least have a few dots on the map to kind of create a kind of a creeked model for what the landscape looked like in terms of its strontium isotope composition. So amongst a whole bunch of other things that would be measured on samples taken from the Bering Land Bridge. So yeah, it's kind of an exciting new project that we only found out about just a couple of weeks ago.
And my gut feeling would be that really on the land bridge, because this must be a big sedimentary plain that was recently covered also by the sea, that the strontium isotope would not have varied very, very fast on this landscape. So that's not really, and I'm not saying that the mammoths did not go on the land bridge, but I'm saying that it did not go only on the land bridge for sure, because we do see like huge variation of strontium isotopes that clearly show big variation within Alaska mainland there. Yeah, that's definitely, again, that's just a gut feelings in terms of what the strontium isotope would look like on the land bridge, but I'm pretty sure it will be the case that it will not vary enormously on that landscape.
Very cool. I have a question from Jenny Granroth, and she would like to know, could these same analysis and statistics be used on the strontium isotope of ancient human teeth and figure out movements of, for example, Clovis people?
I would say that there's nothing about the model itself that's particular to mammoths or mammoth tusks, but I'm not an expert in human physiology. So maybe Beth or someone could speak to that aspect. Can we, oh, Thomas?
I might be able to answer that because we have been working on human migration for the last 25 years in collaboration with some museums. And yes, the answer is yes. I mean, it's one of the I would say, one of the major tools in order to investigate human migration. So measuring the strontium isotope decomposition in the enamel as an archive of the childhood and compare these to the pulp dentine, which is basically the recent information of the last period. And so you're on the one hand comparing if there's a change in the isotope decomposition, and again we're using like isoscapes of regions in order to identify migration, and even worse, so to say, basically a similar approach is used also in human enamel, as the enamel is also growing incrementally. So again, here you see a time resolved resolution and see early human migration pattern as well. And so it's basically the same approach. And funnily, it's you can use it also in hair or in feathers and butterflies. So it's a very unique tool, as a matter of fact.
But the one big difference would be like the lack of the continuous record that you get in the tusk, right? So you would get the beginning, the end, but could you use different parts of the body to kind of fill in those gaps?
Yes. As a matter of fact, like there was one of the mummies found in the Alps, the Ötzi, which where the complete reconstruction took place with respect. You have It's tricky though, because you need to consider the metabolism, and the bone basically stores this information. But in the different areas, with respect of the turnover, you have very recent information, where you have long stored information, and then the early childhood within the animal. Then you have like in the pulp dentine over the last 7-8 years, but in the areas of the bone where you have a lot of stress, like in the long bones where the big muscles attach, and if like it's a big walker, then you have a lot of turnover because you have a lot of mechanical stress. So this is also used in modern osteoporosis. You know, if you do a lot of training, you have a larger turnover, and you can use this time resolution. It's tricky though. So we did an investigation on a sheep. So this got injected a spike, a strontium spike, and after a while we did the investigations. So where did this spike end up at which time? And we found out at the end of the day that the highest turnover of strontium was in the jawbone, because that was basically what the sheep did. It stood there and did chew on grass, so it didn't move too much, so it was chewing, and so there was the highest turnover, which was so the very recent information. And if you compare it to bones like rib bones, where there's little turnover, then you might be able to really give a time record. But this is pretty tricky on the one hand, and the other point is also that the dentine is very porous. So with respect of the repository material, you need to consider diagenesis of these samples, which is not that much pronounced in the enamel, because it is very crystalline and less porous. So there's a lot of things to think about. And at the end of the day, there comes analytical chemistry, and you need to sort out that as well. The other thing is this is also with respect of other investigations. You can combine the strontium like with neodymium. You have lead isotopes also giving you more ideas. And then you have the different ratios of elements, like the barium to calcium ratio, which was indicated before, which gives you an indication of weaning and end of weaning. So there's so much information to read out of this book of life of the different individuals. So it's pretty cool exercise.
If I could add one thing to that, one thing that hasn't come up yet today is that all of the software that we created and all of the analyses that we ran are publicly available, and there's a link to them from the paper. So if you're curious and if you're a little techie, feel free to download them and take a look, and we'd love to hear from you as well.
Also, along those lines of Thomas with the human, I mean, Ben and Jeff might add something about the challenges of dealing with human remains in Alaska.
I think we all need to keep in mind the sort of the differences between using sort of mammoth materials that might be in a museum on federal land versus the very significant issues that come up with human remains. So this isn't the forum probably to talk about this too much, other than to say that it's absolutely essential to have buy-in, collaborative support from the indigenous peoples of the region that are relevant to the remains. And really nothing destructive should ever be done without that close cooperation.
And in addition to that, of course, I mean, the in Alaska, it's the limited number of remains that is a challenge too. I mean, there's
very few, and there's very few in the Americas, just in general, for this time frame. We're lucky to start piecing together some discoveries in Northeast Asia that really have connected the New World and the Old World in sort of interesting ways. But yeah, they're very, very rare.
Thanks, Ben.
Do we have time for questions?
I think we have time for a couple more. the question I was going to ask is from Connie and Ned, and they're asking, is there any correlation in the tusk lines with weather patterns such as drought?
Yeah, well, yeah, that's a tricky question because we can use the oxygen isotopes to get at geolocation, but they could also be reflecting a change in 28 years worth of weather as well, and so we could look at that. So yeah, that's a tricky one.
I could say too that, you know, the little incremental lines, the little lines of von Ebner that reflect, those are daily growth bands, and they averaged around 20 microns in thickness, but they're like tree rings too, and they will vary in thickness depending on seasonal effects. And, you know, drought could be one of those, cold could be another. So that might have influenced the another way of looking at potential weather impacts.
So if patterns were seen, do you see similarities in modern large mammal habitat use? This is from Andy Padilla.
Well, I didn't get the question, sorry.
So if patterns were seen, if you're seeing patterns, do you see similarities in modern large mammal habitat use?
Well, I mean, I guess we don't really have like a habitat map from 17,000 years ago, but what we see in terms of the mobility patterns definitely looks like an African elephant in some ways, you know, just a kind of opportunistic migrants, somebody that really wants to move far away when resources are limited. So there is like definitely similarities with African elephant, I would say, but it would be important at this point to reconstruct also the niche ecology of those animals, particularly looking maybe at carbon isotopes that we do have, that could actually use to build some isoscapes as well for thinking about habitats, but we haven't done that part of the work at this point. So
yeah, and just going back to Ned and Connie's question as well. I mean, part of the information that we gain about the past up here in Alaska is derived from the fossils of animals that were on that landscape. But we also, in terms of like assessing what the weather was like, or the climate was like, or the vegetation was like, that's a huge effort that has been going on, and uses not only the fossils themselves, but also taking cores from lakes, taking evidence from tree rings. I mean, there's ancient DNA from permafrost. Beth can speak to, but that's a whole other industry of fleshing out what were the conditions that these animals lived in as well. And so it's a whole additional science that goes on in what we call paleoecology, which is trying to figure out what past ecological systems were like.
So to end, I guess, our question period, I'm going to ask a Yukon question. And do you have any of the modern Stratium data for the Yukon that could be included to potentially assess if the mammoths from Fairbanks were moving over to Dawson in Canada?
Yeah, so we did, the map that we created did include the unglaciated part of your part of the world as well. And in fact, we're actually, Audrey Rowe is a PhD student here at UAF, has been actually trying to acquire additional rodent samples from your part of the, yeah, you have some?
I have some.
So to kind of add to the mix, to kind of help resolve that part of the world to the map as well. So yeah, that's a work in progress. But we did actually, I think, Clément, we created, or Juliet and you created a map for that unglaciated part of eastern Beringia as well.
When you're doing that road map, you're using the incisors of the rodents, is that, or are you using other parts?
Yeah, generally teeth are good.
And are you just using like the complete tooth, or are you targeting a certain part of it?
We can take a little chip of it at the end. We've done laser ablation on rodent teeth, or Karen and Audrey have done some of that, and we're finding that probably by solution method, so a little chip is like better than laser ablation.
Right.
So with that, I'm going to say thank you so much, everyone, for joining us today. And big congratulations to your paper that came out last week. I don't know if you noticed, we did lose our Facebook feed. So I will say that we will be putting this on Facebook as well, just because we did lose that. And thank you to all our viewers that were able to join. And again, thank you, everyone, for joining us as our panelists.
Thank you for hosting us today. Keshah and Elizabeth, it's been really fun.
What's that? Have a great day, everyone. Thank you.
Thank you.
Care.